Hello guys, today we are going to discuss about types and ultrafine structure of a chromosome. Before going into the topic, we should know some basic parts of a chromosome, right? The central part of the chromosome is known as centromere, right? The terminal parts of the chromosome is called as telomere. These two are called arms, these four are called arms. The one which is long is called as long arm. The one which is short is called as short arm. Right? The chromosomes which are at the same side of the centromere is called as sister chromatids. These both are called sister chromatids. These both are also called as sister chromatids. Right, and this single one is called as chromatid, right? These both are called sister chromatids, and if any other chromosome is there, and this one and this term and this term, these both terms are called as non-sister chromatids. Right, now we'll go to the types of chromosome. First, types of chromosome based on position of centromere. Okay, based on centromere, it's generally divided into 4 types, right? First one is metacentric, submetacentric, acrocentric, and telocentric. First we will study about metacentric. In metacentric chromosome, the centromere is present in the middle of the chromosome, exactly at the middle of the chromosome. This type of chromosome is called as metacentric, right? And the second one is submetacentric. The centromere of the chromosome is slightly above from the mid position, so this type of chromosome is called as submetacentric. This is second case. Third one is acrocentric. If the centromere is present at the tip of the chromosome, like somewhat slightly at the tip, this is called as acrocentric. And in the telocentric, there won't be any centromere, there won't be any arms above the chromosome. Okay, here one important object to make that is arm ratio. Here I will write it. Arm ratio is generally defined as longest arm by shortest arm. This arm ratio will be highest in acrocentric chromosome. Okay, this will be the highest in acrocentric chromosome, not in the telocentric chromosome, because in the telocentric chromosome short arm is not at all present. That's why we will take it as arm ratio is highest in the acrocentric chromosome. Just remember this one. Next, going into the types of chromosome based on number of centromeres. Based on number of centromeres, it is divided again into 4 types. First one is monocentric. If only single centromere is present, then it is said to be monocentric. If two centromeres is present, then it is said to be dicentric. If more than two centromeres is observed, then it is said to be polycentric. Usually polycentric chromosomes we can't observe like so many like this. And the next one is eccentric. Eccentric, as the name states, that centromere is absent. Okay, these are four types based on the number of centromeres. Okay, now let's come to the topic: ultrafine structure of chromosome. For this structure of chromosome, there are so many models, like multi-stranded model, folded fiber model, nucleosome model, like this. So many models are there. In this, today we are going to discuss about nucleosome model, which is widely accepted model. Okay, this nucleosome model is given by Kornberg and Thomas. These are the two scientists who have given this nucleosome model in the year 1974. Okay, this is the universally accepted model for the structure of chromosome. This model generally explains us about how giant is the DNA molecule, and histones are packed. This model generally explains us how giant is the DNA and histones are packed. Okay, let's understand this by some diagrams. Here I have kept some diagrams. First, yeah. Okay. This will let's understand this. See, this is the DNA double helix, right? The size of this will be 2 nanometers, and it will be shrink, and it will form the nucleosome. Okay, now let's understand how this nucleosome will be. Let's understand by this diagram. Yeah, this small this round you can see, right? These are called as histone proteins. Okay, there will be five types of histone proteins: H1, H2A, H2B, H3 and H4. Okay, nucleosome is generally, this nucleosome is there, right? This nucleosome is generally unit of chromatin or chromosome, we can say. It's composed of 200 base pairs, in which 146 will formed by this one, and the remaining will be formed by linker DNA. 75 will be formed by this linker DNA, and 146 will be formed by this histone octamer, right? In this histone octamer, this H2A, H2B, H3 and H4 is present in pairs, and it is, so it will be 2 H2A, 2 H2B, 2 H3 and 2 H4 will be present. And all this, see, for example, this is H2A, H2B, H3, H4, and again this one is again H2A, H2B, H4, H3. Like this it will form the octamer, right? Here you can observe like this all histone proteins will be formed the octamer. This DNA will be condenses and see, this DNA will come like this. So this all will form as the histone octamer because 8 are present, right, in 2 pairs. The H1 histone protein will be lying outside of this nucleosome, right? Like this the DNA will come and rotate 1.75 turns it will take, and it will go outside, and 146 nucleotides pairs of DNA H1 DNA is present in this 1.75 turns. This one you have to remember. And as we have discussed, these are the histone proteins that are generally present: H2A, H2B, H3, H4, H3, H2A, H3, H4, H1A. Like this will be formed the octamer, and this DNA will be coiling 1.75 times here, right? And this is the H1 histone protein. This is the H1 histone protein, right? And in this 146 nucleotide, and in this 1.75 turns, there will be 146 nucleotide pairs will be present, like adenine, thymine, guanine, cytosine, right? Like this 146 pairs will be present. Now let's go into the next diagram. Here let's understand this. Like this double helical structure of DNA will be condensed and form this nucleosome, which we have studied. Like this 6 nucleosome units again will be 1 2 3 4 5 6. Like this 6 nucleosome units again will be condenses and forms this one. This is called like solenoid structure. So this solenoid structure will be forming, and then this solenoid structure will be again condenses and forms this 300 nanometers, and again this will be condenses and it will form 250 nanometers wide fiber, and again this will be more condensing and form this 700 nanometers, and this will condense, and then our chromosome will be forming with 1400 nanometers. Again we will understand using this diagram. See, this is our normal double helical structure of our DNA. This will be condenses and forms this nucleosome, like, and again here this is the beads on a string. See if anything is there, if beads are present like this model is also resembling like that, so it's called as beads on a string form of a chromatin, right? Again this will, like this 6 will be condenses and form this solenoid structure. This structure is called as solenoid structure, and the size of this is 30 nanometers. And again like this so many solenoid structures will be condenses and forms this protein scaffold. This is the protein scaffold. Like this so many nucleosomes will be folded and forms this nucleate like protein scaffold structure, and this will be again condenses and forms this entire, see, Here you can see this small part is this one, like this, so much protein scaffold will be forming, and the size of this is 700 nanometers. So this is the ultrafine structure of chromosome. The H1 proteins are always in the outside, like the nucleosome model of chromatin fiber structure is almost consist of all the evidence accumulated. So this is the universally accepted model. Thank you.